Hello and welcome to this presentation on genetic modification. We will be looking at crops today, and on the left we have corn, which has been genetically modified to contain a gene from bacteria that produces a toxin, and this toxin kills pests. On the right hand side we have some ripe fruit, and genetic modification can be also used to make fruit last longer than normal or to ripen early. So this picture is of golden rice on the left compared to normal rice on the right. Golden rice here on the left has basically been genetically modified to contain vitamin A, also known as beta carotene. And that sounds mostly positive, which it is, because third world countries can lack vitamin A, and it's been the cause of the same number of deaths as HIV or TB in some areas. This modification, though, was not always positive because basically political and financial conflicts meant that this genetically modified golden rice was not transported to the places where it was needed, which ended up in a lot of deaths from children lacking vitamin A and blindness. So this diagram here on the right is of a bacterium. You might wonder why I'm showing you a bacterium, and that's because it's a bacterium that we can put a modified gene into, and then we can infect the rice with the bacterium so that it produces vitamin A in its crop. So how do we do that? So we have a bacterium that provides us with a plasmid, and we cut a gap in the plasmid with a restriction enzyme at a specific point. And on the left, we have the gene of interest. In this case, it's going to be of another bacterial cell. And we cut out that gene of interest. Here it is. This is the gene that contains, with the beta carotene example, it's a gene that causes the production of vitamin A. And it's put inside this plasmid. And then the whole plasmid is put inside a bacterium where it's going to replicate. If you're wondering how we stuck this gene of interest in, it's with a ligase, a DNA ligase. So we've talked about the plasmids, the restriction enzymes that do the cutting, and the ligase that does the gluing. Now, if we wanted a gene from a virus, for example, we would need to use reverse transcriptase to change the RNA into DNA. I wanted to go look at more closely at a plasmid. Now, this is the plasmid that we mentioned on the previous slide. It's cut at a specific point with a restriction enzyme. And this is the gene of interest in green. It's cut with the same restriction enzyme so that it can be inserted into the plasmid. If you look more closely, you can see that the bases are complementary, and this is how they stick together, like magic. And here's the recombinant plasmid again, with the gene of interest in, that's been stuck in with the ligase, and it's put into a bacterial cell. And actually, it naturally is accepted by bacterial cells, so that happens quite easily. And if we want to look in more detail about how the golden rice is made, well, the gene of interest comes from daffodils and bacteria that can make the vitamin A. They are put into plasmids, like we mentioned, and then we infect the seed here with the bacterial plasmids, and they grow into crops which have vitamin A inside. They can be hybridized with crops from the country that you want them to grow in so that they can withstand the weather conditions of this country. And so we've looked at the genetic modification of golden rice so far, and the second video will have a look at some other examples. So see you next time.